Good morning. This is Tuesday, July 28th. This is our last lecture, as a couple people have already mentioned. Absolutely. Last lecture, which is both a good thing and a bad thing. It has been one of the most unique semesters I have ever had. Again, I know it's the summer, it's not really a semester, but last year was kind of crazy because we had half on, half off. But, oh, yes, thank you. Oh, I forgot what I was. So it's been a unique experience doing this whole thing online, and so there have been some bumps and growing pains, but I think overall, hopefully, especially for those of you who are left, I want to thank you for struggling through it, and hopefully it's been beneficial for you. So a couple quick announcements, things I want to talk about first. Again, today is our last lecture. We're going to finish off our autonomic nervous system and start with those pathways. We have the exams Thursday and Friday. Again, many of you, not many, but several of you have gotten a little lax on your scanning. I tried to make sure to make comments on that on the grading so that you'd be aware of that. But again, make sure you are scanning the entire work area, the left side, the right side, behind. I want a whole 360 view of your workspace, not of the ceiling, not of your room. So again, I'm going to be a little more strict on that for these remaining exams. So please do that properly. Please do that carefully. Lab and lecture exam, you know what to expect. Do them in either order. You know about the time that they're going to take, about an hour and a half for the lab exam, about 2.5 hours for the lecture exam. You have all of that. For the final exam, 100 multiple choice questions, 105 minutes to complete the exam. I did add a little extra time so that you guys can do the scanning and all that kind of stuff as was asked. And again, you only have 105 minutes, but you can take it any time during the class time from 8 to 12:35. I'm not just giving you a 2-hour window like they normally do during finals weeks. So you have a little more flexibility with your schedule that way. The other thing I wanted to talk about for the exams, and again, thank you, Ryan, for reminding me, is that I know the final exam seems intimidating. Because again, it's cumulative and it covers everything that you're covering in this class. But if you think about these things from a math standpoint, the final exam is 100 points. Your last lab and lecture exam, your lecture exam is also 100 points. Your lab exam is going to be somewhere between 60 and 70 questions. So there are more points on the nervous system stuff with the lab and the lecture exam than there is on the final. The other thing to remember about the final is all multiple choice questions. So you have all of the correct answers there. You just have to identify the correct answer. With the lab exam, with the lecture exam, you have essays where you have to pull the information out of the ether and describe it. You have identification. The lab and lecture exams are the more challenging type of exams. So while I know there is the onus and of the intimidation of having this huge final exam, it's not that huge in relation to the other things. And definitely your lab and lecture exam is more important, and you should spend more time on that than you do on the final in preparing for that. The other thing that I want to remind you about for that is, like I said, be careful how you're studying for the final exam. Again, I know it's easy to get bogged down in all of the information because you go back and you look at all the muscles, all the origins and all the insertions and all the actions and all those things. And is it possible that one of the multiple choice questions on the final exam could be what bone does the humerus originate from or something? I mean, does the bicep brachia originate from? Sure, absolutely. Would it benefit you to study the origins, insertions, and actions? Sure, absolutely. But if you think about it, we've had something like 15 chapters in this section, 14, 15 chapters in this section or something along those lines. So on average, there's going to be about 7 questions per chapter, right? Again, and so some chapters were a little bit smaller, like the chemistry, maybe only 4 or 5, but where others like cells, maybe there'll be 8 or 9, but there's going to be around 7 questions per chapter. It's going to be spread pretty evenly through all the material. So spending half of your day on Wednesday just looking at origins, insertions, and actions isn't necessarily a good use of your time. Right? Again, one of the things that we've seen in this class is all the information builds on itself. For that final exam, 50% of the information should just be kind of aha information. Things we've used so much in this class, you know, like that the most, you know, the most common cation in the outside the cell is sodium. The most common cation inside the cell is potassium. right? The sodium potassium pump moves 3 sodium out of the cell and 2 potassium into the cell. You know, there are some pieces of information we've used again and again in this class, and those questions are there because, again, that's the point. We're building on this stuff and we're building a foundation that is going to help us in 431. You don't get to just forget this stuff at the end of this class. You can't just mind dump. The whole point, the same way that I haven't randomly picked the information, we've used the information as we move forward, we're going to continue to use this information into 431 as well. When you get to the respiratory system, when we talk about the nasal cavity, that it has a ciliated pseudostratified columnar epithelial tissue, you're going to know what that means. Right? So again, it is going to be important to do this. The final exam is important. You do need this information moving forward, but your lab and lecture exam are equally important, if not more important. So make sure you're using your time wisely. All right, excellent. And Ryan, thank you again for that because I had totally forgotten, even though the note's right here in front of me. All right. Any other questions on any of that or anything else before we get started? All right, excellent. So let's go ahead and pick up where we left off. Let's look at the pretty picture from the textbook first. As we mentioned, we are going to be talking about our sympathetic pathways. And here we see, you know, what a three-dimensional illustration of this. Again, the only thing that's really lacking from this picture is, as we know, up here, in the front of the vertebral column is going to be those unpaired collateral ganglia. And the reason I'm not showing it is because technically it's down a little lower than it would be in this view. Because we see the ribs, it would be down below the ribs. So that's why they haven't shown that there. But these are going to be the types of pathways. Notice one of the things that I want to emphasize as we look at it, as we look at this, and I'll highlight it here. This chain ganglia, you see there are many pathways, 1, 2, 3, 4, 5, and there is actually a 6th that this picture doesn't show coming off of it as well. So we're going to need to talk about those when we work through that. Here we remove some of the bones and stuff like that, and we see most of the components of this. Again, we don't see the 2 additional pathways that are coming off of this, one there and one there, but we will talk about those. And then this was the picture that I found online that most kind of accurately represents, and again, as you can see, it was a very small picture that I've blown up, and so it's kind of fuzzy, but it does remind us of the basic anatomy, which we're going to draw again, and hopefully you already have your handouts prepared for this. We have our spinal cord, we have our ventral root, our dorsal root, they come to the spinal nerve. The spinal nerve, again, notice we're missing the dorsal ramus, but we have the ventral ramus, we have the gray ramus, we have the white ramus to our chain ganglia. And notice this chain ganglia finally is a chain ganglia that has 1, 2, 3, 4, 5, 6 pathways coming out of it. And that's really what we want. We'll talk about the chain ganglia again. We're going to beat this dead horse as much as we possibly can to make sure that we understand it. And so that's our goal. As we talked about, we need to, and so let's go back to here. We need to be able to identify all of the sympathetic pathways. That is way too huge. Black, and let's start with 18 for that. All of the sympathetic pathways. Again, on the exam, I'm going to give you any of the possible effectors for our sympathetic nervous system. So anything our sympathetic nervous system can affect, from the top of our head down, from erector pili muscles to salivary to lacrimal glands, to the nasal mucous membranes, to the salivary glands, right, to on and on down, heart, lungs, spleen, stomach, pancreas, bladder, everything, any of those. And you are going to need to know the pathway and be able to describe the pathway to them. The good news is that there are basically 4 main pathways. Oops. Wow, that's not a 4. That we'll take. There are some subtle differences in them, and we'll talk about those subtle differences as well. But there are 4 main pathways that we can take with our sympathetic nervous system, and we are going to identify all of those. Now, I'm going to draw them out. Obviously, you do not need to draw them on the exam, so you'll just need to write out the driving directions. And that's really what we're doing here. We're giving driving directions. If you were trying to give someone directions to your house, you could draw them a map of how it is, or you could write it out in words. I'm going to draw you the map. You're going to write it out in words when it comes to the exam. So I will show it to you visually. You will write it down appropriately. So let's start with the basic anatomy. As we mentioned, we are going to start, because all of our sympathetic pathways start in the spinal cord. We need to start in our spinal cord. You'll need four of those drawings. Although you could get away with 3, but 4 would be ideal. And I'm going to take a minute to draw it here. So if you need to quickly draw yourself another one, that will work as well. Let's move this just a teeny bit more over this. I want as much room as I possibly can. There we go. That works. And then let's draw our gray horns. Excellent. We know coming out up here at the top, we have our dorsal root with our dorsal root ganglion, even though it's not going to play any role in this path, and our ventral root to our spinal nerve. And then from our spinal nerve, we have our dorsal ramus. That can go up this way. Whoa, no, there. We have our ventral ramus that goes off this way. We have our chain ganglion down here, and we have 2 paths to that. The lateral ramus, which is the white ramus, and the medial ramus, which is the gray ramus. And I'll get rid of my lines in just a minute. We know that this is part of a chain, so I'm going to cheat, and even though it lines up in a straight chain, I'm going to go ahead and draw my chain, cheating a little bit by going off that way and then going down this way. So this goes to a second chain ganglion, which would be down here. Actually, no, hold on. I want to undo that. I do actually want to switch this off to the side. So here, so again, I'm not lining my chain up, even though we know the chain should be lined up. This will just allow me to cheat a little bit on this process. And again, obviously those aren't drawn to scale, but again, hopefully you are getting the idea. And then I need 2 more paths going out, this one here and this one here. Excellent, so 6 paths going into and out of the chain ganglia. But remember, the chain ganglia, those paired chain ganglia, are not the only ganglia. We also here in the front have our collateral ganglia. All right, so now we've got all of that. That is the drawing that we need. Let's go ahead and label everything that we have talked about. We're not going to label everything yet. We're going to add a couple labels along the way. Let's label the things we know. The things we know, and for this, I'm going to write it as small as possible. The lateral gray horn. Our ventral root. We know the dorsal root, but like I said, since this is a motor pathway, it's not a part of this path, so I'm not going to bother labeling it. This here is our spinal nerve. This here is our dorsal ramus.
I'm sorry, the spinal nerve, is that one going sideways?
Yeah, this one right here. Remember, the ventral root and the dorsal root come together to form the spinal nerve. So the spinal nerve is this part right here. Can you see my mouse? This part right here is the spinal nerve. Remember, the spinal nerve is the part that actually comes out the intervertebral foramen. And then as soon as it comes out the intervertebral foramen, that's when it branches to form the 4 branches. Ventral ramus. Excellent. White ramus. Gray ramus. These are, where should I put it? I'll put it right here. Chain ganglia. As is this a chain ganglion. As is this a chain ganglion. Because remember, these chain ganglion are all bound together. Hold on, see, there was another question. The posterior gray horn was for sensory, right? And the lateral gray horn was for what again? Lateral gray horn, remember, is where we have our autonomic motor. Anterior gray horn is somatic motor. Posterior gray horn is sensory. And lateral gray horn is where we have our autonomic motor. So that is where all, again, if you think about it, our sympathetic pathways all start in the central nervous system, all start in the lateral gray horn. So for all four pathways, we all are going to begin at the same location. Yeah, so we're all starting in the same location, and then it will change a little from there. Now, obviously, these chains are connected. Some are above and some are below. So this here would be the descending chain. And then this up here would be the ascending chain. Excellent. Going to the chain ganglia above, going to the chain ganglion below, and then lastly, this is our collateral. Ganglion. Excellent. All right, I can move that out of the way. I can hide this down here for now. Perfect. All right. So there's my drawing. A little bit smaller than what we had before, but that is a good thing because then what it will allow me to do over here, and the first way we're going to describe when we talk about the four main pathways, we are going to identify them by the effectors that are innervated. So basically, which effectors are we going to go to, and what are the paths that we're going to get there? All right, and like I said, there's basically four main pathways. All right, everybody comfortable with our starting point? All right, everybody's completely silent, so that means yes. All right, so remind me again. Oh, and here, there's the one last thing that I wanted to cheat and do. I want to open up my pathways. There, that looks a little prettier. And let's screenshot that. All righty, excellent. So let's start with the chain ganglia. Where are the effectors? What are the parts of the body that we said would be innervated by, would be connected to the chain ganglia? What do the chain ganglia go to? There are basically two destinations that we said the chain ganglia would go to. Anyone remember where we said the chain ganglia innervated? I'll wait if you need to look back at the notes. You have to give me at least one of them. Skin, excellent, perfect. Skin is absolutely one of them. And what's the other? Organs above the thoracic cavity. So that includes the head as well. So internal organs above the thoracic cavity. Excellent. Perfect. Those are indeed our two potential effectors from our postganglionic neurons in the chain ganglia. So our path 1 is going to be to the skin. We did skin first, so let's go to the skin. As we mentioned with the skin, we are going to synapse. And now this is too big, so let's switch back to that. In a chain ganglion, right? Any one particular one, or pretty much all of the chain ganglia, right? So we really don't have to discriminate. All of the chain ganglia have the potential to innervate the skin by synapsing in the chain ganglia. So that's going to be our goal. We need to synapse in our chain ganglia, and then we have to get out to the arms, to the legs, to the trunk, to the skin. Now, as we mentioned, there's always going to be 2 neurons in this pathway. This is a 2-neuron pathway, and we'll make neuron 1 red. I think that's what I've been doing. I don't honestly remember. So neuron 1, of course, is our preganglionic neuron. And tell me what we know about our preganglionic neuron. What is some of the information we know about preganglionic neurons? Excellent. They have a myelinated axon. What else? True, they're going to synapse in a ganglia. And now, so we know where it ends. Where does it start? Excellent. Cell body in the lateral gray horn. Excellent. Perfect. So then let's draw that. We are going to start with, we forgot the one last piece of information. If I'm going to draw this, what kind of style am I going to draw? What is the structural classification of this neuron? Multipolar. Excellent. You'd think after a semester and a half of this, I'd be a better typist, but you would be wrong. Excellent. So, multipolar neuron whose cell body is located in the lateral gray horn, and it is a multipolar neuron. Now, as we know, when its axon leaves the spinal cord, what structure does it enter into? Dorsal root, ventral root. Out the ventral root. Excellent. From the ventral root, it goes to the spinal nerve. Excellent. Now, at the spinal nerve, it has choices, 4 different destinations it could pass through. Now, its goal is to get to the chain ganglia. So that means it's probably going to form or go through either the white ramus or the gray ramus. And that brings us back to the other piece of information we have on our preganglionic neuron. Its axon is myelinated. And so if you're an axon that is myelinated and has this myelin sheath on the covering of it, do you think you're going to form a gray ramus, or do you think you're going to form a white ramus? White ramus, exactly. So from the spinal nerve, our axon forms the white ramus and enters into the chain ganglia. And throw a little more myelin on here, just as that reminder. Excellent. Now, this is our goal. We want to synapse in our chain ganglia. So we have reached our destination. However, I will remind you that, remember, we said our sympathetic neurons branch elaborately. So while its axon will synapse right away, will likely synapse right away, it's also very likely to ascend and form some synapses above, or to descend and form some synapses below. It's going to form hundreds of synapses. Some at the level where it enters, some can ascend and some can descend. But we're just going to focus on one synapse. That one synapse is going to be on our postganglionic neuron. There's its cell body there. But before we do that, remind me again what neurotransmitter my preganglionic neuron is going to release. Excellent. Releases, and I'm going to go ahead and abbreviate it too, even though we do want to spell it out if we were doing this as an essay question on the exam. And what is the effect of it? Is it excitatory or is it inhibitory? Actually, I lied. I will write it out. And it is excitatory. Excellent. So then we can cheat right here. Right, ACH for being released, and put a big positive there. Bingo. We are done with our preganglionic neuron. We've done half of our pathway. Now, before we can finish our pathway, we need some information on this postganglionic neuron. What do we know about it? Multipolar. What else? Unmyelinated. I'm going to try to put the stuff in the same order so that it looks similar. Oops. Unmyelinated. Excellent. Axon. It obviously starts here in the chain ganglia. Well, let's say in a ganglion. In this case, it happens to be the chain, but it could also be the collateral. And this is going to end where? Effector. Excellent. At effector. The structural classification is multipolar. What neurotransmitter is it most likely going to release? Most likely releases norepinephrine. And again, it's either going to be excitatory or inhibitory depending on which effector it ends up at. All right, excellent. So we can draw our ganglia, pardon me, not our ganglia, our dendrites on our postganglionic neuron. So there's some dendrites we have going on there. Excellent. We have our dendrites. Now, our goal is to get to the skin, to get superficially, to get out to the dermatomes of the body. Do we have sensory and motor neurons already going to the dermatomes of our body? Like the muscles and skin of our back, like the front of our body and our limbs? Do we have some axons that are already going to my finger, for instance? To move my finger, to feel my finger? Yeah, how would they get, how do nerves leave the anterior gray horn, get to my finger to make my finger move? What of the structures that we have labeled here would we use to get to my finger? Any idea? Which one of these nerve structures formed our dermatomes? Do you remember? No, not a bad guess. No, remember, it's the dorsal and ventral rami. Remember, the dorsal ramus carries axons to the back, the skin and muscles of the back. The ventral ramus carries nerves to and from my arm and the front of my body. So those are already structures going to my finger or going to my back. So if I want to control the erector pili muscle of my finger, I need to get back out to that ramus. So to get to the skin, we need to get to the dorsal and ventral ramus. So it's going to travel out. So, oops, I don't want that one to be black. If I'm going to travel out my dorsal and ventral ramus, I have two choices I can take. I can take the white ramus to get to the dorsal and ventral ramus, or I can take the gray ramus. And seeing as I am a postganglionic neuron, and what do we know about the postganglionic neuron? Its axon is unmyelinated, so there you go. Because it has an unmyelinated axon, that unmyelinated axon travels out the gray ramus, and then either goes out the dorsal ramus to the skin and muscle of the back, or it can go out the ventral ramus to the skin and muscles of the front and limbs. And of course, as we mentioned, and I'll just have to write it up here in between, it is going to release norepinephrine, and depending on the effector, it can be excitatory or inhibitory. And there you go. Just that simply, we have described our first pathway. So notice, if I said a sudoriferous gland, this would be the pathway you would describe. And with one last piece of information, on the sudoriferous gland, do you think that that norepinephrine would be excited? Do you think the neurotransmitter that it releases would be excitatory or inhibitory? If you're sending a sympathetic signal, are you going to sweat more or are you going to sweat less? More, so it's going to be excitatory. Excellent. If it's the erector pili muscle, excitatory or inhibitory? Excitatory, makes the hair stand up on end. There you go, exactly. Right. However, if it was, for instance, the blood vessel going to the muscle, that would be inhibitory because we would want to dilate, we'd want to relax that muscle, dilate the smooth muscle of that blood vessel and bring more blood to the muscle. So in that case, it would be inhibitory. So depending on the effector, you should be able to tell me whether it's going to be excitatory or inhibitory. All right, this is how we play the game. Any questions on this? Because if this one doesn't make sense and doesn't work, it's just going to get worse from here. All right, excellent. So, nope. Collateral ganglia is connected, but it's not used in this pathway. So yes, so you are correct. When the time comes, I can extend this line, and because this is going to connect to it. But since the collateral ganglia isn't part of this pathway, I didn't bother doing that yet. And because we haven't identified that structure yet, I was going to wait for that as well. But yes, eventually we will connect it. For the skin, correct. For the skin and for the muscle, right? Skin, and then let's, in parentheses up here, skin and muscles. Make that black. Excellent. All right. Skeletal muscles. Let's be more specific. Or actually, let's be really more specific. Blood vessels to the skeletal muscles. There you go. So that would be the same pathway. Thank you for asking that, Madison. All right, so that is path one. Any other questions? I love these questions. Like I said, I want to make sure this makes sense because if it doesn't make sense, then we've got trouble. Okay, excellent. Then I think we are ready for path two. Path two, as we mentioned, are internal organs above the diaphragm. Okay. That means the organs of the head, That means the organs of the thoracic cavity. All right, so we're talking about two different things here, organs of the head and organs of the thoracic cavity.
Is this a second path for a second picture?
Yes, this will be for a second picture, although we'll see it's not a completely entirely, but yes, second path, second picture. Okay? Now, as was mentioned, where do we want our synapse to be? Where are we going to synapse if we're going to go to the internal organs above the diaphragm? Chain ganglia or collateral ganglia? Chain ganglia. However, as we know, unlike skin, where all of our chain ganglia go to the skin, do all of our chain ganglia go to the head? Well, I'm asking the question, so the obvious answer is no. So let me ask the question I really want the answer to. Which of our chain ganglia by itself, essentially, because again, there is overlap, but by itself basically innervates all the organs of the head? Which of our chain ganglia does that? Okay, partial credit for cervical chain ganglia. How many cervical chain ganglia do we have? Three. Do all three of them innervate the head? Nope, just one. There you go. Excellent. So the only chain ganglia that innervates the organs of the head is the superior cervical chain ganglion. Oops, doesn't quite fit in, so I'm just going to go gang. There you go. Oops, also might help if I spell superior correctly. Excellent. Actually, you know what? I have to do that anyway. Excellent. So, which specific chain ganglia innervate the organs of the thoracic cavity, like the heart and the lungs and things like that? Middle and inferior. Excellent. Cervical chain ganglion. Excellent. So now in this case, when we're describing our pathway, we can actually specifically describe it, which specific chain it is that we are synapsing in. All right, now obviously I need to carefully try to clear some of this so that we can get rid of that. Get rid of that, get rid of that. Now, I could continue to try to very carefully erase the rest of this, but do I need to? Our goal is to synapse in a chain ganglia. Isn't this what this path shows? Yeah. So notice the beginning, really everything that the preganglionic neuron does in path 1 and path 2 are identical. So we are going to start with a preganglionic neuron with a myelinated axon whose cell body is located in the lateral gray horn and wants to synapse in the chain ganglia. So it starts in the lateral gray horn and it is multipolar. Its myelinated axon travels out the ventral root to the spinal nerve, to the white ramus, to the chain ganglia, although now we can be specific. If we're going to go to the head, and I'll just abbreviate it here. It's going to be superior cervical chain ganglia. And if it goes to the thoracic cavity, then your answer would be the middle and inferior cervical chain ganglia. So again, depending on what destination I give you, you would want to identify the specific chain ganglia as to which one it is going to be. But it is then going to synapse on this chain, and just like before, it is going to release acetylcholine, and that acetylcholine is going to be excitatory. So look how convenient that is. In paths 1 and path 2, what the preganglionic neuron does is essentially identical. All right? However, where the two pathways are different is where our unmyelinated axon of our postganglionic neuron goes. Again, this time our destination is an organ like something in the head, like the eye, like the nose, like the heart, like the lungs, etc. To get to that destination, what our postganglionic neuron does is its axon, its unmyelinated axon, leaves out of, and I'm going to cheat, give myself a little bit more room to there. It is going to leave out the chain ganglia via a different structure. That different structure that these postganglionic neurons axon forms is what we call the sympathetic nerve. So our axon, instead of going out the gray ramus, goes out the sympathetic nerve, and it goes to the eyes or the nose or the heart or the lungs or any of those organs above the diaphragm. Of course, as we talked about, it is going to release, oops, make that blue again. And cheat again, give myself a little bit more room. It is going to release norepinephrine, most likely, and it can be positive or negative, depending on which effector it is influencing, right? If it goes to the heart, it's excitatory. If it goes to the lung, it's inhibitory because it's dilating the smooth muscle, letting more air in. If it's the eye, it is excitatory, dilating the eye out. If it's the nose, it's inhibitory, drying up your mucous membranes. So again, depending on the effector, it's going to be positive or negative. And there you go. There is path 2. Questions on that? All right. Just that simply, we have completed path 2. That brings us to path 3. Path 3, whoops, I want that to be black, is going to be the internal organs below the diaphragm. Now, for these internal organs below the diaphragm, where are we going to synapse? Does this synapse in our chain ganglia? No, collateral ganglia. It's going to synapse in collateral ganglion. There we go, that works. How many collateral ganglia are there again? How many collateral ganglia? 3, absolutely. 1 of 3. What are their names? Excellent. Celiac. What's the next one? Superior. Oops. Mesenteric. And inferior mesenteric. Perfect. Excellent. Remind me one more thing. Which internal organs below the diaphragm did we say the celiac ganglion innervated? Remember, we said it was the proximal digestive, but we want to be more specific than that. What were some of the organs? Excellent. Oh, I like that. All right, let's see. Liver, gallbladder, stomach, spleen, pancreas. Excellent. Perfect. Excellent. There you go. What does our superior mesenteric innervate? There you go. Small intestine and the proximal large intestine. Excellent. And then hopefully you figured out our third question. What is it that the inferior mesenteric ganglion innervates? Excellent. So distal large intestine, or again, you could just say rectum and anus. Because after all, who doesn't want to say rectum and anus? Excellent. Urinary system and reproductive system. Excellent. So again, in describing the pathway then, when we're over here and talking about where our synapse is occurring, should we be able to say, if I say that it is going to, that I want you to give me the pathroom to the liver, then will you need to say specifically that it's the celiac? Or if I say specifically that it's going to the small intestine, you need to say that it's the superior mesenteric ganglion. Or if I say it's going to the bladder, do you need to be able to say that's the inferior mesenteric ganglion? Yeah, absolutely. So you should be able to know which collateral ganglia specifically we are synapsing in when you are describing these pathways. All right. Now, we need to reset our picture then, obviously. So we need to get rid of this. In fact, we know we're not synapsing in the chain ganglia. So we need to get rid of all of that. And we need to get rid of our synapse here. But before I do any more erasing, what I will tell you is that we don't need to erase anymore. Because, let me cheat with that so I can get this a little closer to here so we know we're talking about the chain ganglia there. One of the complaints that people always have about Siri when it's giving driving directions, or back in ancient times, it wasn't that. You would go to, before you went someplace, you didn't have your Garmin, you didn't have your phone, you didn't have Siri, you didn't have Waze, or all of those types of things. So before you left the house, what you would do is you would type into Google Maps where you wanted to go, and they would give you 5 pages of driving directions. And the first 3 pages were just getting out of your neighborhood, right? Everybody knows how to get out of their neighborhood. So everybody's saying you could have start at step 6 after I leave the neighborhood. Well, that's basically what happens here. The starting part of our pathway is going to be the same. We are dealing with a preganglionic neuron whose cell body is still in the lateral gray horn of the spinal cord. Its axon still has to come out the ventral root because that's the only place it can go. It still has to go to the spinal nerve because it's the only place it can go. And again, dorsal and ventral ramus take us to the surface of the skin, the arms and the legs. We don't want to go there. It's going to be the same for path 4 as well, absolutely. It is only one place it can go, to the white ramus. And that white ramus is going to bring it to the chain ganglion. However, this time we do not want to synapse. This time we just pass through without synapsing. So this time our axon is going to continue. That myelinated axon is going to continue through the chain and out. It is still myelinated. And it is going to travel all the way. That looked horrible. It still looks horrible, but it serves our purpose. And because even though I'm incredibly messy, I'm also very anal. Let's get that. There we go. Excellent. Our myelinated preganglionic axon travels all the way to one of the collateral ganglia. Which one depends on which organ we are interested in communicating with. Again, if it's the stomach, it would be the celiac. If it's the proximal part of the large intestine, it would be the small mesenteric. If it's the distal part of the large intestine, it would be the inferior mesenteric. Whichever one of the 3 it is, there it is going to synapse. And as we know, there it is going to release acetylcholine, and it is going to be excitatory. So now we are synapsing in a collateral ganglion. And of course, we are synapsing on a postganglionic, no, that was supposed to be blue, on a postganglionic neuron, who we know is multipolar. Whose axon leaves, oh, I guess I need to draw another, hold on, so let's undo that. It is going to form and leave out of a structure. Not surprisingly, it is an internal postganglionic neuron. It is a sympathetic nerve, just called a sympathetic nerve, just like the ones above the diaphragm. These are sympathetic nerves below the diaphragm. And its myelinated axon, unmyelinated axon is going to leave and go to the effector. And again, anything from the stomach to the bladder, to the small intestine, etc. And of course, when it gets there, it is going to release norepinephrine, and it will be excitatory or inhibitory. depending on the effector. And there you go. We have given our driving directions to our third potential type of destination, some internal organ below the diaphragm. All right. Questions on pathway 3? All right, excellent. Hopefully that makes sense. Let's go ahead and save that pretty picture, make sure it makes some sense to us. And that brings us to pathway 4. So of course, my question to you is, what the heck is left? We've gone to the skin and muscles of the body. We've gone to all the internal organs above the diaphragm, and we've gone to all the internal organs below the diaphragm. What the heck is left? Anyone have any idea what other special route our sympathetic pathway may need to make? Somebody, I think it was Ryan, actually pointed it out on the very first lecture of this, like 2 minutes in. When we were looking at that octopus-looking picture, somebody noticed that there was something weird going on with the adrenal gland. And that's actually our fourth destination. Our fourth destination is the adrenal gland. The fourth possible place that our sympathetic nervous system wants to innervate is the adrenal gland. And remember, the adrenal gland sits on top of the kidney, so it is also sometimes referred to as the suprarenal gland. So both of those are acceptable terms. So let's talk about the pathway here. Now, in some ways, this fourth pathway is much more like a parasympathetic pathway, because with our adrenal gland, the synapse actually occurs inside the organ. So we're not synapsing in a chain ganglia, we're not synapsing in a collateral ganglia, we are actually synapsing on a ganglia that is inside of the organ. So if you think about it, that is kind of like our parasympathetic pathway, where we had those terminal ganglias inside. However, to get there, we do pass through the celiac ganglion. So we're not going to synapse there, but we are going to pass through the celiac ganglion. So let's reset our picture a little bit for this fourth pathway. And I'm going to cheat because I'm lazy. First, we need our destination. Here is our kidney. Oops, no, hold on. Let's make that black. There is our kidney. And then sitting on top of our kidney is our adrenal gland. And yes, I appreciate this is not drawn to scale. However, it does let me do a little bit of funny work here. Our adrenal gland is one of those organs that has a chewy nougat center and a candy-coated shell. We have seen that type of anatomy before. When we have this type of anatomy with an outer region and an inner region, what did we call the outer region again? Cortex, excellent. And the inner region is known as the medulla, excellent. I'm putting the medulla out here where I can draw a line to it, because the medulla is actually going to be our destination. Our true destination is the medulla of the adrenal gland. All right. So, because I'm incredibly lazy and a horrible artist, and also because it would be hard to do, I'm not going to bother erasing my pre-ganglionic neuron, because the good news is I don't have to. The path starts the same way. Our preganglionic neurons multipolar cell body is located in the lateral gray horn of the spinal cord. Its myelinated axon leaves out the ventral root, enters into the spinal nerve, passes through the white ramus to a chain ganglion, where it does not synapse. It passes through without synapsing, forming our, I didn't name the structure on the last one. Dagnabbit. This structure that it forms when it leaves is the Splanchnic Nerve. Remember how I told you in our parasympathetic we had those pelvic nerves, which are also called the pelvic splanchnic nerves? Well, this nerve right here that is made up of these preganglionic myelinated axons that travel from the chain ganglia to the collateral ganglia are known as the Splanchnic Nerve. So let's write this up here to remind us. Through the Splanchnic Nerve. So I need to move that a teeny bit. Excellent. So again, on 3, we go through that splanchnic nerve to the collateral ganglion synapse. Here on pathway 4, we go through the splanchnic nerve to the celiac ganglion and pass through without synapsing. So, oops. Undo. Red. Still myelinated. Out that sympathetic nerve. And as it leaves out the sympathetic nerve, it travels to the medulla. And finally, synapses in the medulla. And of course, when it synapses in the medulla, it releases acetylcholine, and it is excitatory. And it does that on our postganglionic neuron. Our postganglionic neuron is a multipolar neuron located here inside the medulla of our adrenal gland. And what it does is our postganglionic neuron releases epinephrine and norepinephrine into the bloodstream. So it releases its chemical signals into the bloodstream. And so if you think about it, with our adrenal gland, what are the effectors of this 4th pathway? Well, once that epinephrine and norepinephrine gets into the blood, where can it go? Everywhere, exactly. Everywhere. So basically everything, right? This provides our short-term hormonal response to stress. And let's be more specific, provides our global short-term hormonal response to stress by releasing that adrenaline into our blood. And that, oh, no, wrong button. And that is our fourth pathway. I would say identify the pathway by which the sympathetic nervous system innervates the adrenal gland. All right. There you go. Those are our four possible pathways. And notice, as we said, those four possible pathways take us to every single possible effector in our body. Everything that the sympathetic nervous system innervates, you would use one of these four pathways to get there. For a couple of them, you may need to identify the specific chain ganglia or the specific collateral ganglia. But other than that, the four pathways don't change. There's one way to get to the skin and the blood vessels of the muscles. There's one way to get to the organs above the diaphragm, one way to get to the organs below the diaphragm, one way to get to the adrenal gland. And one of these, I guarantee you, will be an essay question you will have on the exam. All right? Questions on that?
Yeah, I got a question.
Yeah.
When I'm looking at the drawings, it looks like only the first path goes through the gray ramus. Now, is the gray ramus melanated or it's unmelanated? Because I know the white ramus is melanated.
Right. So remember, what helps to form the gray ramus are the unmyelinated postganglionic neurons axons. So since the ones that pass through this are unmyelinated, they're leaving, they're postganglionic, that's why it's the gray ramus.
Got you. Okay.
Yep. All right. Any other questions? All right, we are going to go through this like 3 more times in different ways, and of looking at this information. But this was the heavy lifting, and so let's give a few minutes for this to sink in. Let's go ahead and take our first break. It's 9:10 essentially, so let's come back at 9:25, and at 9:25 we will pick up the lecture from here.
One more question.
Yes.
Are they all multipolar?
Yes, both preganglionic and postganglionic neurons are autonomic motor neurons. So functionally, they are autonomic motor. And as we know, all motor neurons are multipolar. So yes, every single neuron in all of these pathways, and again, there's only 2, but all the neurons, so all 8 neurons in these 4 pathways are all multipolar.
Okay.
All right, so like I said, we will restart at 9:25, and I will start the recording at that point. Any other questions? All right, I'll see you in 15 minutes. So we've gone through all of this with the pretty pictures. Let's go through these steps again in the words, so that again, because that's how you're going to be describing them, to make sure that we understand this. Now, again, there are lots of ways you can make sense of this information. As we just finished talking about, we focused on the effectors and then how to get there. But again, another way we can think about this is, where is the synapse taking place, right? So that's one of the ways that we can think about these processes as well. And as we talked about, one option would be to synapse in a chain ganglia. And if you synapse in a chain ganglia, again, as we put the pieces of information again, what are the possible effectors that you could end up at if you're going to synapse in a chain ganglion? Excellent. One would be the skin, or organs above the diaphragm. Excellent. Excellent. So, all right, we leave the preganglionic neuron. We start with the preganglionic neuron, I'd say, whose cell body is located in the lateral gray horn. Its myelinated axon leaves via the ventral root to the spinal nerve. We should probably have that in there, because that's the only place it can go. But then from there, oops, hold on, I don't like that. To the spinal nerve. From there, it goes down the white ramus, which again is the lateral communicante, where it will synapse on that chain ganglia. Again, because these things branch elaborately, some of it may ascend, some of it may descend before it synapses, but it is definitely going to synapse in that chain ganglia. And from there, our postganglionic unmyelinated axon is going to exit out the gray ramus, the medial communicante, going to the dorsal ramus or the ventral ramus. And as we mentioned, this path is the one that takes us to the skin. Or the smooth muscle of the blood vessels, of the muscles. All right, something along that route. Conversely, we can also synapse in the chain ganglia, take that exact same route, starting in the lateral gray horn, out the ventral root, spinal nerve, white ramus, synapse within the chain ganglia, or ascend or descend, because remember our second path is exactly the same as the first. What's different about the first and the second is what happens to that postganglionic axon, the unmyelinated axon. In this case, it forms our sympathetic nerve, and it goes to the thoracic cavity, right? And then from there, it either goes up to the head, or the heart, or the lungs, right, etc. Basically, everything above the diaphragm. Oops. All right. Now, as I mentioned, your book does do a nice job of illustrating these. And so it has given us some pretty pictures that will help us to remember this. So here we see, this one isn't actually from your textbook, but I like this picture a lot because it shows both sides. It shows the collateral ganglia, and it also shows us the medulla, the adrenal gland, its cortex and medulla that we have here. And notice what we see here are those two possible pathways. Now, again, I want to emphasize on this illustration, they're showing one path on one side and one path on the other. It's not like all the sympathetic nerve paths are on the left and all the gray ramus paths are on the right. It's paired, it goes both ways, but they're just showing them separately. Again, notice for both these first two pathways, what happens with that preganglionic axon, what happens with that preganglionic neuron is the same. Starts in the lateral gray horn, ventral root, spinal nerve, white ramus, synapse. Release acetylcholine, excitatory. Lateral gray horn, ventral route, spinal nerve, white ramus, synapse. And notice this time it shows that it can ascend. Synapse, release acetylcholine, be excitatory. So the difference between the first two pathways is what happens with our postganglionic neuron. If its axon goes out the gray ramus to the dorsal or the ventral rami, then it's going to go to the skin, go out those dermatomes to the skin and to the muscles. If instead its axon leaves out that sympathetic nerve, then it's going to those visceral organs above the diaphragm, heart, lungs, eyeballs, salivary glands, etc. All right. Questions on those two? Very similar pathways, the first two. Again, the only difference, two differences, where the postganglionic axon goes and what the effector is. And again, most are going to release norepinephrine and be excitatory or inhibitory depending on the effector. So those first two paths are very, very similar. And here's the picture from your textbook showing this. Again, notice here it is showing our route to the skin and to the blood vessels like we talked about. And your book also makes a point of emphasizing that it can ascend or descend. So that, again, notice here it enters the chain, ascends before it synapse, but it's still going to go out the gray ramus and go out to the skin. All right. Questions on those first two routes? All right, excellent. Then let's talk about route 3. Again, this time where we're synapsing, because that's what we're focusing on, the difference here, synapsing in a collateral ganglia. Now, as we talked about, the route begins the same way. Our preganglionic neuron, cell bodies located in lateral gray horn, out the ventral root, through the spinal nerve, to the white ramus, to the chain ganglion. But this time it does not synapse within the chain ganglia. It leaves the chain ganglia without synapsing and forms the Splanchnic nerve. That Splanchnic nerve goes to our collateral ganglia, or what we call the prevertebral ganglia. And there it's going to synapse in one of our three collateral ganglias: celiac, superior mesenteric, or inferior mesenteric. And which of those three it is going to be determined by which effector it is. Remember, the celiac goes to the proximal digestive, so liver, gallbladder, pancreas, spleen, stomach. Superior mesenteric goes to the small intestine and the proximal large intestine. And the inferior mesenteric goes to the distal large intestine, the reproductive organs, and the urinary organs. So that unmyelinated postganglionic axon leaves the collateral ganglia, and it doesn't say it here, so let's remind us. It forms a sympathetic nerve as it leaves and innervates basically all of the organs below the diaphragm. Again, we have some nice pictures that show this as well. This is the one not from your textbook, but again, it shows both sides. And what I like about this is this really emphasizes that the chain ganglia are paired, whereas the collateral ganglia are not. So notice preganglionic axons from both sides go to that collateral ganglion, synapse in that collateral ganglion, and then out those sympathetic nerves to whatever organ in the abdominal pelvic cavity that it is.
Question?
Yes.
When does it become unmyelinated?
So the postganglionic neuron's axon is unmyelinated. So the black one here is unmyelinated. The preganglionic neuron's axon is myelinated. So anywhere that this preganglionic neuron goes, it's myelinated the entire path. It has, this one has a myelinated axon, this one has a unmyelinated axon.
Okay, so it stops being myelinated when it reaches the collateral ganglion, correct?
The pathway is, but again, I would be careful in wording it that way. You have two different axons with two different characteristics. So it's not like the preganglionic axon gets halfway and then becomes unmyelinated. The preganglionic axon is myelinated. The postganglionic axon is unmyelinated. So you have two different axons with two different characteristics.
Okay, gotcha.
Yep. All right, excellent. I get a kick out of this picture from your textbook. This is the picture from your textbook, and I will give them a little bit of credit for trying to hedge it. So let's just look at the picture first, and it makes sense. Lateral gray horn, ventral root, spinal nerve, white ramus, through the chain ganglia without synapsing, through the splanchnic nerve, synapse in the collateral ganglia. Postganglionic leaves that through a sympathetic nerve and goes to an organ. However, notice which organ they picked: intestine. Now, first of all, they didn't even specify whether it was large or small intestine. But notice the collateral ganglia they picked is the celiac. Now, notice they did say such as, they did say for example. But did they pick matching ganglia and organs here for their examples? Does the celiac innervate any of the intestines? No. So again, they did, I guess, kind of hedge their bet a little bit in that, but it was a weird decision to do it that way. But so yeah, so yes, ironically, your picture is from your textbook. While they show an absolutely correct pathway, picked crappy names to put in here for these. Maybe just celiac was smallest, so they didn't bother to put, superior mesenteric because it took up less space. And someone really liked drawing the small intestine. I don't know, whatever the reason, but ironically, it's different. But at least the pathway that they show is correct. All right. Excellent. Our fourth pathway, or again, we are thinking of in terms of where the synapse takes place. The fourth pathway, the difference here is our synapse takes place in the medulla of the adrenal gland, or the suprarenal medulla. Again, path starts the same way. Pre-ganglionic neuron starts in the lateral gray horn, out the ventral root, part of the spinal nerve, through the white ramus, to a chain ganglia, where it does not synapse, out the splanchnic nerve, and that definitely we want to make sure we put out here. Out the splanchnic nerve to the celiac ganglia, where we don't synapse in the celiac ganglia either. Instead, we leave the celiac ganglia, out the sympathetic nerve, to the medulla of the adrenal gland, where it forms its synapse. And that postganglionic neuron, it still synapses on a postganglionic neuron, and it is the postganglionic neuron that is going to release the hormones into the blood. About 80% of what it releases is epinephrine, and about 20% is norepinephrine. Madison, you have a question?
Yeah, because I put it in the comments, but I think we were all typing too fast. Will we have to say which specific collateral ganglion that the postganglion is going to?
Yes. If
Okay.
I give you if I gave you a specific destination, like
Would you
the
say
stomach,
specifically the small intestines kind
right?
of thing?
So if I say what is the pathway to the small intestine, then you need to say superior mesenteric. If I say to the liver, you need to say celiac. If I say to the bladder, you need to say inferior mesenteric. So yes, for that pathway, there's only three, but yes, for any of those third pathways where it goes to the organs below the diaphragm, you absolutely need to specify which collateral ganglia it is. The same way, if you're going to an internal organ above the diaphragm, you need to say whether it is the superior cervical, the middle cervical, or the inferior cervical chamber.
Okay.
So yes, if
just wanted to clarify that.
So yeah, if it's an internal location, you have to say the specific ganglion. If it's just the skin, then you can just say chain. For that, you can just say chain and don't have to be specific.
Okay,
But any internal organ, you have to say the specific ganglion. Yep, great question. Yes, thank you for that. All right. And
question.
again, yes, go ahead.
Yeah, this might be like a stupid question, but like the
The only
paths.
stupid question is the question not asked.
Okay, so like the path 1, path 2, path 3, and 4. I mean, we just chose the numbers, but they can be either one, right? I mean, we're not gonna have a question like, what is path 4?
You are correct. I am not going to ask the question, describe path 3. That is not going to be a question.
Gotcha.
I will, like I said, the way the question will be worded is it will be something along the line of, describe the sympathetic pathway to X. And X is going to be some effector. So it could be a sudoriferous gland in the skin, an erector pili muscle, the sweat glands, the salivary glands, the tear glands, your heart, your lungs, your stomach, your spleen, your gallbladder, your small intestine, right? The testes, the anus, any of these locations. I will give you a specific effector, and you will need to describe the pathway. So again, yeah, the numbers that we've given these are completely arbitrary. But the ultimately, if you think about it, there's practically 100 questions I can ask. But with all those 100 questions I can ask, there's really only 4 pathways you have to describe. So yes, based on the effector, you will need to know which of these 4 pathways it is, and you will describe it. Okay?
Yep.
Excellent. So again, as I mentioned, your book does describe this 4th pathway to the adrenal gland, but it doesn't give a picture. That's why I mentioned, remember, I had the 3 at the beginning and a line through it. It doesn't emphasize it the same way. Luckily, I do have that pretty picture from a different textbook that shows this. So again, notice this clearly shows our pathway. Pre-ganglionic neuron starts in the lateral gray horn, out the ventral root, to the spinal nerve, down the white ramus, through a chain ganglia without synapsing, out the splanchnic nerve, to the celiac, even though they don't have it written here. We definitely want to write it here. This is the celiac ganglion. Without synapsing, to the medulla of the adrenal gland, where it finally synapses on that postganglionic neuron. And that postganglionic neuron is then the one that releases the adrenaline into the blood, where it goes everywhere. All right. Now, notice when we come back to our octopus, now that we are more sophisticated, we can actually see how, as I mentioned, this really does a nice job of showing this. Notice here is path 1. Path 1 is starting in the lateral gray horn. Preganglionic neuron leaves that, and now we know it's forming the ventral root, the spinal nerve, the white ramus, to go to a chain ganglia, where it is going to synapse in that chain ganglia. Once it synapses in that chain ganglia, our postganglionic neuron goes up the dorsal and ventral root to go to the blood vessels, the adipose, the erector pili muscle, the sweat glands of the skin. There's our first pathway. Pathway 2, starting in the lateral gray horn, ventral root, spinal nerve, white ramus to a chain ganglia. Notice in this case, it's going to need to ascend to be able to get to the superior cervical chain ganglia, where it synapses. And then that postganglionic neuron goes out a sympathetic nerve to the eyeball, or to the salivary glands, or something like that, something in the head. Notice the middle and inferior, same thing. Synapse in a chain ganglia, postganglionic goes out the sympathetic nerve to the lungs, to the heart. Path 2. Path 3, start in the lateral gray horn, ventral ramus, ventral root, spinal nerve, white ramus, chain ganglia, pass through without synapsing, forming our splanchnic nerves. This splanchnic nerve goes to the celiac ganglion, where it synapses, and goes to the liver, or the gallbladder, or the stomach, or the spleen, or the pancreas. Or we can leave the spinal cord through the chain ganglia, splanchnic nerve to the superior mesenteric ganglia going to the small intestine or the proximal large intestine, or we can go through the chain ganglia, synapse in our inferior mesenteric ganglion, and go to the reproductive organs, go to the urinary system, go to the rectum and anus. And notice this even has our fourth pathway. Start in the lateral gray horn, ventral root, spinal nerve, white ramus, pass through the chain without synapsing, form in the Splanchnic nerve, pass through the celiac ganglion without synapsing, forming that sympathetic nerve, traveling all the way to the medulla of the adrenal gland, where it synapses and releases adrenaline into the blood. So while this was a very scary picture when we first looked at it, we can see that this picture actually does a really good job of showing us all four of those pathways. So there's really a lot of meat on this illustration. Yes?
Like we have, you know, all the organs here, like the lung, the heart, and all that. Is this all the organs that we need to know for the exam, or is there other organs that are not covered in this picture that we need to know?
Well, I mean, I guess there are some other things, like for instance, the large blood vessels of the thoracic cavity would, for instance, be located up here. And again, it would still be the same pathway of going from the middle or inferior to here. I guess here we did talk about it's going to the eye, but this is also going to go to the lacrimal gland, right, where we cry. So there may be a couple minor organs that might be, but I think that those would, I think like for instance, those two are definitely obvious ones. The lacrimal gland is obviously in the head. The large blood vessels are obviously in the thoracic cavity, are obviously in the thoracic cavity. So you should be able to infer the pathway. But I think this is pretty much everything. There are, you know, maybe a few minor things, but this is the meat of it. Yes, absolutely.
Okay.
Yep. Yeah, I think those are the only really the big two that that aren't jumping out at me right now that should be on here that really aren't. But again, I think that hopefully those two pathways would be obvious. Nothing else jumps out at me as being missing.
And we also have to describe where it says the cervical sympathetic ganglia. We do have to break it down into superior, middle, and inferior, correct?
For these three right here, absolutely. These three right here, if the pathway is going above the diaphragm, then it's going to be one of these three cervical, and you need to specify which cervical chain ganglion it is, yes. The superior just goes to the head, middle and inferior just goes to the heart and lungs. Okay? Yep. All righty. Hopefully this is helping. If not, then let's beat the dead horse some more. Again, one of the other ways we can think about this is where our sympathetic preganglionic neurons are going, the different routes that it can take. Because again, remember, it all starts the same. That preganglionic neuron starts in the lateral gray horn. It has to come out the ventral ramus. It has to go to the spinal nerve. It has to go to the chain ganglia. But once it gets to the chain ganglia, it has choices. It can, one, synapse right away. It can, two, ascend or descend first before it synapses. It can continue through and synapse in a collateral ganglia, or it can continue all the way through to the adrenal gland, which obviously isn't here on the picture, but is the fourth thing then that preganglionic neuron can do. Again, while we're beating dead horses, probably one of the last things that I want to do to emphasize this, and this is also a question that makes a good essay question, and let's take a look at this. What you can see here with our chain ganglia, one of the things we've mentioned is that our chain ganglion basically has 6 structures going into or out of it. And these are the major pathways, the highways and byways. So for each of these for each of these 6 passages, we need to identify them. We need to say whether they're formed by pre-ganglionic axons or postganglionic axons. And of course, if we are identifying that, then that also tells us if it's myelinated or unmyelinated. So we have to identify them. We have to say if they're formed by preganglionic or postganglionic axons. We also need to know the direction they carry information. Right? Are they carrying that? No, that's not how you spell information. Are they carrying it into or out of the chain? And if it's going out, what's the destination? If it's coming in, where did it come from? Right? So these are all pieces of information we should be able to identify. And again, this helps us with our driving directions. If we know what's going on with the chain ganglia, we can really kind of figure out everything else that is going on with it. All right? So let's do this. Let us do this, because again, this is another great essay question. Save this one last time, although I think I already have, and then I can clear this all. And let us go ahead and draw a chain ganglion. And actually, I'll make it small because we need more room actually around it to write than we actually need for the thing itself. And I'm going to go ahead and draw our 6 pathways into them. And despite my strong, strong desire to be anal and open up these pathways, I'm just going to leave them attached to the circle like this. At least for now. It may eventually bug me and I may fix it. But we'll see if I can keep my OCD at bay and just have it look ugly. It's hard to be a horrible artist and anal at the same time. It's a curse. All right, excellent. So there are 6 structures coming into and out of a chain ganglion. Give me one of them. Identify one of these 6 things coming into or out of the chain ganglion. Give me the name of any one of them. Splanchnic nerve, excellent. Let's, for argument's sake, make this one the splanchnic nerve. I'm going to need to make that a little smaller. Excellent, we'll save white Ramus for next. I like that one. We'll do that one next. So, that's a good size. Excellent. So let's answer all those questions. Splanchnic nerves, are they formed by preganglionic or postganglionic axons? Post, excellent. No, not post. Preganglionic. It is preganglionic axons that form our splanchnic nerve. So does that mean they're myelinated or unmyelinated? Myelinated, excellent. Are they going into the chain or out of the chain? Out of the chain. And if they're going out of the chain, which direction are they going? Where are they going to? What is their destination? Excellent, going to a collateral ganglia. Excellent, that's simple. And these are preganglionic neurons, and I think we've been using red, so let's stick with red to remind us that there we go. We have those splanchnic nerves. Preganglionic preganglionic axons that are myelinated, carrying information out, going to the collateral ganglion. Excellent. I think, let me jump back. I think the next one that someone mentioned was the white ramus. So let's go ahead and next do our white ramus. And let's answer those exact same questions for the white ramus. White ramus, preganglionic neurons or postganglionic neurons? Preganglionic neurons, excellent. So if they're preganglionic neurons, myelinated or unmyelinated? Myelinated, excellent. Coming into the chain or out of the chain? Into the chain, excellent. And if they're coming into the chain, they're coming from? Where did it start? Where's the cell body located? Spinal nerve, I like spinal nerve is a good answer. Spinal nerve is a good answer. That is definitely the immediate thing. Or the other way we could have said it is where it ultimately started, and that is the lateral gray horn. I like both of those answers. I would absolutely accept both of those answers. So I like that. That is perfect. Excellent. So that is that white ramus. And again, this is preganglionic. So that's red, myelinated, going in. Excellent. All right. Someone also mentioned the gray ramus. I saw that as the third one that someone mentioned. Gray ramus, preganglionic or postganglionic? Pre, post. All right. I have one pre, two posts. It is indeed post. Postganglionic axons. So does that mean it's myelinated or unmyelinated? Unmyelinated. Excellent. Excellent. Going into the chain or out of the chain? Out of the chain. And if it's going out of the chain, where is it going to? There you go. I like that. It's going to the dorsal ramus or the ventral ramus. Absolutely, that is the immediate location it's going to. Or, again, we can think of its ultimate destination. It's basically going to the skin.
I got a question.
Yes.
So I'm looking at all the drawings, and every drawing that I'm looking at from path 1 all the way to path 4, from what I understand, it should be the post. I'm sorry, the pre-ganglionic, not the post. Can you explain to me?
The gray ramus?
Yeah.
Here, hold on. Let's go back to this. So let's go all the way back here. So notice, this is our sympathetic pathway. Notice it is the white ramus where the preganglionic neuron comes in. And if we are going to go to the skin, it's synapses in the chain ganglia. And then it is the postganglionic, that blue neuron, whose then unmyelinated axon then comes out the gray ramus. The only thing that comes out the gray ramus are these postganglionic unmyelinated axons as they go out, in this case, to the ventral ramus to go to the skin on the arm, for instance. So the white ramus, because again, think of it in terms of their color, white and gray. The white one is white because it contains myelinated axons, and the preganglionic neurons are myelinated. The gray ramus contains unmyelinated axons, and the postganglionic neuron, that's the one whose axon is unmyelinated.
Okay. Thanks.
Yep. Hold on. I'm sure I can do this. Let me see if I can figure this out. Oops, not that one. Huh. I don't seem to have my drawing of the first pathway. That's disappointing. I'm not sure how that happened. But here, let's see if I can share this. I thought I hit it like 18 times. Maybe it'll show up after I close this. Maybe it's not there yet. I don't know. It's weird. Here, if we look at our drawing here, notice the words are still here. So if you remember for our first pathway, and I'll make this green just to make it different. Remember, this postganglionic neurons, unmyelinated axon, came out the gray ramus, where it would either go out the dorsal ramus to the skin and muscles of the back, or it would go out the ventral ramus to the skin and muscles to the front and the limbs. So the only structure that comes out that gray ramus is that unmyelinated postganglionic axon.
Yeah, so it looks like all four paths use the white ramus to go in, and only one of the paths uses the gray ramus to get out.
That is correct.
Okay.
All right, excellent. Perfect. All right, we're boom, halfway there. So give me another structure that either enters into or comes out of the chain ganglion. Sympathetic nerve, perfect. Sympathetic nerve. Is the sympathetic nerve made of postganglionic or preganglionic neurons? Or axons, I guess, more specifically. Excellent. So, of course, does that mean myelinated or unmyelinated? Unmyelinated. Those two answers go hand in hand. Excellent. Going into or out of the chain ganglion? Out of. And it's going out of the chain, going where? Where is it going to? Excellent, I like that. So we could specify organs above the diaphragm, internal organs above the diaphragm. Oops. Or we could be more specific, right? Head, heart, lungs, right? Stuff like that. Excellent. So again, need to draw our arrow now, and that arrow is coming out. Excellent. So there's 4. That leaves us with 2. What are the 2 that remain? Excellent. Perfect. I like that. Absolutely. This up here would be the ascending chain, and this one down here would be the descending chain. So we have our ascending chain and our descending chain. We'll do the ascending chain first, although really the answers to these are going to be identical. Are the ascending chains made up of preganglionic or postganglionic axons? Which ones were the ones that we said could go up and go down? Pre, excellent. Preganglionic axons, excellent. So of course that means they are myelinated, excellent. But here's the tricky question. Are they going into or out of the chain? Here you go. I like both answers because both answers are correct. It goes both into and out of the chain. Because if you think about it, what's going to happen is that one axon coming in here could ascend to the chain above, but one from the chain above could descend and come down. So it's actually going to go in both directions. But remember, whether you ascend or descend, you are still going to synapse in a higher or lower chain ganglion. So they're not going to leave the chain. They're just going to a higher or lower chain ganglion to synapse. So in this case, we are red, and it is going in both directions. And the same thing is happening down here. The descending chain, because again, the ascending chain of one chain ganglion is the descending chain of another one. So here with our descending chain, it's going to be identical. Pre-ganglionic axons that are myelinated, that go both into and out of the chain, and they're going to synapse in a higher, lower, chain ganglion. There you go. So that is our other potential sympathetic pathway question. Notice here with our chain ganglia, we were able to identify the 6 structures that came into it and out of them. Identify what forms those structures, whether it's preganglionic or postganglionic axons, whether they're myelinated or unmyelinated, whether they go into the chain or out of the chain, and if they go out, where's their destination? If they're coming in, where did they come from? So again, notice there are lots of ways that we are capable of processing this information and hopefully making sense of it. We've talked about the individual. I did, yes, thank you. I just did save this one. I made copies of all the other ones. I unfortunately don't have that first pathway. I will post these pictures, absolutely. However, remember, you can't just recreate these pictures on the exam. You need to write out in words what it is that they're doing and saying, but I do want you to have these visuals, so I will. And I don't know how I did not save that first one, but I am unfortunately missing the first pathway. But at least on the second pathway, as I showed you, it does still show the destinations. So again, if we sneak a peek at that picture again, if I still have it up. Where was it? Yeah, here it is. So again, like I mentioned, while I didn't save the picture, we still have here the description of it over here. And like I said, all you'd have to do, again, hopefully you've been drawing this as you've been going along as well. But again, all you need to do is remind yourself that that first pathway is this one here, where it's going to go to one of the, out the gray ramus to one of these 2 structures to go to one of the 2 parts of the body. So this now shows both the first and the second pathways on it. Okay, so it's hopefully a pretty easy fix. So I apologize. I have been trying to save the pictures. I'm not sure what happened with the first one. I'm hoping maybe when I close everything out, it'll be there. So maybe something weird happened. So we'll see what occurs with that. All right. So let's go back to the lecture. All the way back to where we were. Here you go. And again, your book's got some nice illustrations showing these things as well, because again, we haven't truly beaten this dead horse enough. Again, all starts in the lateral gray horn, that preganglionic neuron. And if you think about it, there are 3 places it can synapse: a chain ganglia, a collateral ganglia, or the medulla of the adrenal gland. If it synapses in the chain ganglia, it goes to the skin, it goes to the visceral organs above the diaphragm, goes to the collateral ganglia, it goes below the diaphragm, and if it goes to the medulla, it basically releases that hormone into circulation and affects everything. And again, if you're getting out to the spinal nerves, the gray and white rami, You go into the skin, erector pili muscles, blood vessels of the skin, blood vessels of the skeletal muscle, those dermatomes. Sympathetic nerves from a chain ganglia go to the heart, lungs, esophagus, thoracic blood vessels. Splanchnic nerve goes to everything below the diaphragm. And again, remember that splanchnic nerve will also take you to the adrenal gland as well. So yeah, I think hopefully I have provided you with as many different ways of understanding this information, because I appreciate that you got the test 2 days from today. So again, I really wanted to try to hit this as hard as we could, to try to hopefully make some sense of it to help you to be successful in 2 days. All right. So again, now we're back to this pretty picture that we saw from before, where we were talking about all the different pathways. And notice again, even in the simple illustration, we can see that it goes all the way to the adrenal gland. There isn't a second neuron there because the second neuron is inside of this. And so like we said, these octopi that we've been looking at actually do a pretty good job of showing us these basic pathways that we have talked about. All right, excellent. So the last thing that I wanted to mention is, notice as we are coming out of the ganglia and forming those sympathetic and parasympathetic nerves, they combine together along with ganglia to form elaborate networks. And what is that fancy term we use for an elaborate network of something? A plexus, absolutely. These plexes typically lie along major blood vessels, major arteries, and so that's where they get their names. These include, and again, you don't have to worry about writing down the names of these. I'm not going to hold you responsible for all of these, but here are some examples of them. And here's the pretty picture where we see this in the body. Again, notice, and I'll use my highlighter to emphasize these. Here are the chain ganglias that are going down one side and coming up the other. So those paired chain ganglia. And notice if you look closely, here along the midline is my celiac, here is my superior mesenteric, and here is my inferior mesenteric collateral ganglia. So we can see where those ganglia are located there. But these are also where some plexes are located as well. And notice if we remove the pesky person, it's a little easier to see some of these plexes and these elaborate networks that are formed by the nerves. However, like I said, I'm not going to hold you responsible for all of these plexes. So no, a plex, a great question. A nucleus in the central nervous system is analogous to a ganglion in the peripheral nervous system. Because remember, nucleus and ganglion are both clusters of cell bodies. A plexus is an elaborate bundle of nerves. Now, nerves and tracts are comparable things. A bundle of axons in the central nervous system we call a tract, where a bundle of axons in the peripheral nervous system is a nerve. But if you put enough of these nerves together, you get this big, elaborate mesh-like structure that we call a plexus. As I mentioned, I'm not holding you responsible for all of these, but there is one plexus I did want to point out to you. Here, where the celiac ganglia and superior mesenteric ganglia are located, this collectively forms what we call the celiac plexus. You may not have heard the term celiac plexus, but maybe instead you've heard it by its other name. Its other name is your fourth or fifth chakra, or what is also known as your solar plexus. How many people have heard the term solar plexus before? Anyone? Heard of it? Someone's heard of it. Excellent. A couple of you might have, might not have. Well, you might not have heard of the solar plexus, but maybe you've experienced this before. You happen to be standing by the soccer field, flirting with some cute guy who's hanging out there playing. And while you're talking to them, right, maybe you do a little stretch to show off some of the body. And as you're doing that, someone kicks a soccer ball and it hits you right below the ribs. You were unprepared for that, so you weren't able to tighten up the core of your body. And what ends up happening to you? Do you die because of that? Now, you're not going to die from it, but you're going to feel like you want to die, because what happens when you get an unexpected blow immediately underneath the rib cage? You get the wind knocked out of you. Absolutely. You have trouble breathing. The reason you have trouble breathing is because one of the structures that comes down here and forms part of the celiac plexus is your friend and mine, the phrenic nerve. And what did that phrenic nerve control again? What does the phrenic nerve control? The diaphragm, absolutely. Remember the phrenic nerve was that nerve that came off the cervical plexus, right? So it was way up here protected, but it goes all the way down to control the diaphragm, and it goes through this celiac plexus. So what happens is when you get that force of unexpected pressure in the abdominal pelvic cavity right below the diaphragm, that force disrupts the activity of the celiac plexus. And in disrupting that activity, you lose the ability to control your diaphragm. Now, you don't die from it, thankfully, because as we know, we have accessory organs that are able to help to elevate the lungs and allow us to breathe, right? Holding your hands up in the air helps when you've had the wind knocked out of you, because it helps to elevate the ribs so that you're able to breathe okay. So like I said, you feel like you're going to die when you get the wind knocked out of you, but you don't actually die. And the reason you get the wind knocked out of you is because of that disruption. That increase in internal pressure disrupts your celiac plexus or your solar plexus, disrupting the activity of the phrenic nerve, and you can't control your diaphragm for a few minutes. So there you go. So again, may not have heard of the celiac plexus, probably haven't heard of the celiac plexus, may not have heard of the solar plexus, but most of you either had the wind knocked out of you or have seen someone get the wind knocked out of them. And so that's why it occurs. All right. Questions on that? All right. Here is the good news. Yes, solar plexus and celiac plexus are interchangeable terms. Notice the celiac ganglion is in the center of all those axons. And so that is why this plexus and the axon have the same name, because they're right next to that big blood vessel. But the ganglion is the cluster of cell bodies. The plexus is formed by the nerves, but they're all together around that and work together. All right. So the good news is we are done with most of our heavy lifting for the day. We still have some more information to go over, but almost all the information we're going to be talking about now is not new information. We will be synthesizing information we already know. We may be adding some additional labels to things, giving fancy names to things that we already know, but there's not a lot of new information. But like I said, we've done a lot of heavy lifting. So I think even though it's been slightly under an hour, although it's been pretty darn close, I think it's time to take our last break. And then when we come after this last break, we will whip out the last bit of information. And then we are finally done with our discussion for Biology 430. So let's go ahead and take a break. I show the clock as 10:21, so let's come back at 10:36. And at 10:36, we will put it to bed. So, you think after all the typing I've done, I'd gotten better at it, but nope, I have not. All right. Any more questions before we take our break? All right. I will see you guys then in 15 minutes. All right. So, everybody's still here. That's awesome. So like I said, at this point, I have said everything that I can think of that you guys need to know, but this is your opportunity to ask me any questions. If you guys have anything that's unclear, if there's anything I can help you with to master some material and help you to be more successful over the next couple of days, I'm happy to answer any questions that I can. So yeah, so what I would say is that, and again, this is not going to be precise, but in general, I would say that about 50% of the lab exam is going to be the central nervous system, anatomy, and histology, right? So again, those are things like the cephalons. Those are things like the motor neurons, Purkinje cells, all of those kind of things that's on that one handout, and the histology handouts. I would say it's going to be about 5% cranial nerves, and the anatomy associated with that, and then about 25%, I don't know why I keep hitting 25% of the autonomic anatomy. So again, things like, you know, the celiac ganglion, or the, you know, spear cervical ganglion, or chain ganglion, or the white ramus, or things along those lines, all those anatomical things. I could show you some of those pictures and point at that. And so I think in general, that's going to be the breakdown. Now, again, obviously there's some overlap on this. The lateral gray horn is part of the central nervous system anatomy and part of the autonomic anatomy. The ventral root is part of the autonomic anatomy and the central nervous system anatomy, things along those lines. But I would say that in general, that's a fairly good breakdown of how it's going to fall out, about like that. So for the lab exam. Okay.
I got a question.
Yeah.
I know you mentioned a few possible essay questions. Is there any way you can maybe repeat some possible essay questions that might be on the exam?
Well, so again, here's what I would say. One of the things that I like about this class is that it's not tricky. It's hopefully very straightforward and obvious. So what I would look at are what are the big concepts that we spend a lot of time on. Those are the things that are going to be likely essay questions. What are like some of the lab type activities, right? So like the Labster, or PhysioX. You know, those types of things. Those are things that easily could be potential essay questions. I definitely mentioned that one of our 4 sympathetic pathways are going to be an essay question. Action potentials is probably the biggest physiological process. So needing to describe, right, comparing an action potential to a graded potential, producing an action potential, propagating an action potential with myelin, propagating an action potential without myelin, right? Those are 4 big concepts that we talked about, and one of those could easily be on the exam. You know, part of the challenge in answering this question, is not everybody's going to get the same questions, right? Because again, the way the randomness of it, everybody's going to get different questions. So normally when I write an exam, I write one version of the exam and everybody gets the same version of the exam. That's not going to be the case here. So everybody's going to get different questions. So I can't just say this is going to be what's going to be on the exam, because it's not as simple as that. But definitely those big concepts that we spend a lot of time on, or we've used multiple modalities to discuss, are the things that are going to be on there. But there's also going to be smaller things as well. Again, if you worked through what we talked about today, just take a lecture like we did today and you go through it, you can find all of the possible, or most of the possible essay questions. I already told you sympathetic pathways are going to be one of them, right? Or the chain ganglia, into and out of the chain ganglia. Right after that, we talked about the solar plexus. So obviously that wouldn't be a big essay question, maybe a 2 or 3 point essay question, or maybe a multiple choice question. So probably not an essay question. But after that, we talked about receptors. We talked about the different types of receptors. We talked a bit about the different types of neurons, you know, cholinergic and adrenergic. And then we finished off talking about receptor agonists and receptor antagonists. Those were the big concepts we talked about today. And so those are the things that are all going to be possible essay questions. Now, are all of those going to be on your exam? No. But again, because of the randomness, any of those are possible questions that could be on your exam. So any of the big concepts we talk about during the class, and that's the nice thing about having the lecture slides. By having the lecture slides, you can go back through your lecture slides and see, all right, what were the big concepts? What were the big processes? Because that's the other thing too, where you have to describe steps, where something has steps to it, or where there are descriptions to things. Those are things that always make good essay questions. So as you're looking through the material, we really had to describe that. That's a good possible essay question. Here was something that had 5 steps to it. That's going to be a good essay question. You should be able to look through this material and pick it out that way. Because quite frankly, that's how I make the exam. I look back at my lecture slides and go, all right, what did we talk about on this day? Well, this is a good essay question, this is a good essay question, this is a good essay question. And that's kind of pretty much how I make the essay questions. That's how I make the exams. So you should study for them in the same way, right? The textbook is a great resource to help you to understand the information. But the lecture slides are what we are talking about in class. The lecture slides are things that I think are important. And since I'm writing the exam, the things that I think are important are going to be the things that are on the exam. So look at the lecture notes, see what we talked about, see the things that were big concepts, and those are the things that are going to show up on the exam. And for this section, for whatever reason, there have been more of those pictures, so I have been posting them. Again, I do apologize. I don't know how I didn't get a picture of the first. Like I said, I'm hoping it will magically appear when I when we end this and convert the lecture, but I'm fearful that it isn't there. But at least you have the material there, and it's close enough to the second one where you should be able to figure out pathway 1 from that. And again, you also have your lecture slides that show it as well. So again, you don't have to rely on my pictures. And lastly, again, you don't want to be memorizing the pictures anyway. The map is useful, but you're going to get driving directions. So you need to write out with words the driving directions. So just having that and having the lecture to go back to will help for that. All right.
Question.
Yes.
After we finish the exam on Friday, the final, when can we expect to see our actual grade for the class? And also the people that were going on for the Wednesday for the extra crediting, you're gonna add that at the end of the class, correct?
So yeah, great questions. Yes. So you are correct. I have not gotten, so let's talk about Open Lab. Jeff has not provided me with the hours yet for the Open Lab. He's actually in Oregon, or he was. I tried to contact him this weekend, and he was in Oregon. Although my understanding is he is doing Open Lab tomorrow. So I believe there's an Open Lab tomorrow he will be doing. But he's in Oregon right now. Typically at the end of the semester, he sends me those hours, and I put them in. And I purposely don't want them from him early because I don't want students relying on that. That is extra credit that can help you, but ultimately I want you to get the grade that you want without it. So get the grade that you want without it. Then if you fall a little bit short, you have the potential extra credit from the Open Lab. Remember at the beginning of the semester, I told you all that you got 15 points of participation points. Remember, those participation points are not extra credit. It is a grade, 15 out of 15. But anybody that is still here listening to lecture, you know, the 16 of you who have taken all the exams and are still around, you guys are all getting your participation points. You all have done a great job with that, so you're getting the full points of that. And again, I purposely don't put that in the grade book because I don't want you relying on it. But if you get close, these things can bump you over the top. So at the end of the semester, and again, it's really going to depend on how long it takes me to grade. It's been taking me about a week. At least we don't have lectures to worry about, so I'm hoping to get it done sooner. My hope is by Monday or Tuesday, I will have all the grading done. What I will do at that point is I will post the full completed grades onto Canvas. At that point, I will post those so that you can see your grades, you can see all your points, you can see everything that is there. Then you do ultimately have a decision at that point, because I know for some of you, a C is a big deal, right? A C grade is a passing grade. Remember, as we've talked about, you can take a class like this up to 3 times. A C grade is considered a passing grade, and if you pass a class, you cannot take that class again. At any Los Rios school. And I know grades matter for most programs, so a C is tricky. A C does allow you to go on to, true, C's do get degrees, but C's don't always necessarily get into nursing programs. That's the problem with C's medicine, depending on which kind of program you're getting into.
I was using that lightheartedly.
No, but you are correct. C's, yeah, you know, that's what they say. What do they call the person who gets the lowest grades at med school? Doctor. right? So yes, you are correct. So the C's do get degrees, absolutely. But again, grades do matter for getting into programs. So the problem we have is this. If you knew your lab and lecture exam score, and you knew going into the final exam that you weren't going to be able to get a B in the class, you would have the choice to not take the final. And if you didn't take a final, you get a 0 on a final, and that would drop your grade significantly. But because the tests are back-to-back, there's no way for you to know this. And I'll be honest, the whole reason they don't let you take the class again is strictly financial. The school gets money based on the number of butts in seats. How many butts are in the seats is what determines how much money the school gets. And the problem is if you've had a passing grade and you take that class again, you don't count as a butt in the seat. And so while they used to allow, there were all sorts of different ways. Heck, my very first year at a Los Rios school teaching, I was teaching anatomy and physiology, and there was a student, and again, you got to remember, we're talking like 15 years ago. There was a student who was taking the class for the 7th time. Because not only could you take it to 3, but she was able to get some other waivers and some other, there were some other rationales and some other things that happened in her life where she was able to continue to get more and more waivers to be able to take that class. And she was taking it her 7th time. That's not possible anymore. The most you can do is take things 3 times now because of the impactedness of this class. And with a passing grade, technically there is a waiver form you can fill out to try to get the ability to take the class again. But since the, economy crashed, what was it now, 6-8 years ago that happened, and especially now with coronavirus, I haven't seen one of those be approved since then. So because it is strictly administrative, what I am willing to do for you is to go back retroactively and remove your final. So what I will do is I will post the grades. Like I said, I'm hopeful. I am hopeful to post the grades Monday or Tuesday. So we'll see there. Hopefully Wednesday at the latest, but I'm hoping by Tuesday would be the latest that I would be able to do that. But I will not make the grades, I will not make them official until at least Friday. So that will give you 3 days to look at your scores. If there's any questions on any of your grades or any concerns or anything along those lines, you can ask that. And ultimately, if you decide that a C doesn't help you in your long-term goals, then what you can do is you can ask me to remove your final, and that will drop your grade to a D. And if I drop your grade to a D, that allows you to take the class again. So if you get a C and you want to take that C and you want to move on, great. More power to you. Hopefully you'll get the grade you want and it won't matter. But unfortunately, every semester there's 2 or 3 people who don't get the grade they want, but want that opportunity to improve it. And so if you fall into that small category, then what you need to do as soon as I post the grades, contact me, let me know to drop your final. I will drop your final, I'll drop your grade to a D, and that will allow you to retake the class again. Because again, if we had more time between the lecture and lab exam and the final, that could be a decision that you made on your own. This is not something that I've decided to do as a wild hair up my butt. I have talked to the dean about this. The dean is 100% on board with this and understands the rationale behind it. So this is something that I can comfortably do, which is why we're talking about this, heck, why I'm still recording right now. So again, if you fall into that category where you get a C and don't want that C, I can drop your final to bring your grade down to a D so you can retake the class again without having to worry about trying to get that waiver cleared. Because quite frankly, it won't. All right.
Sounds good.
Excellent. All right. Any other questions?
Yeah, quick question. Did you say that there's no study guide or anything for the final, correct?
I do have a study guide, but that study guide is really for the final in class, and the online final is slightly different. So there are some slightly different topics in it that aren't necessarily covered on that. And there was one student last semester, at the end of last semester, because we were kind of half and half, that was upset by that because it was and was confused by that. So I'm not using that right now. So like I said, what I would encourage you to do is to use your lecture notes. Know that every single chapter we've covered, right, from introduction to chemistry, every single chapter is going to have somewhere between 5 and 7 questions on it. So it really is going to be spread throughout all the material. So no,
Okay.
I don't have a study guide for you for your final. Nicole's question, the 3-page histology handout plus the hour lecture. Yes, absolutely. I would say that that falls under, that is going to fall. So the histology handout is part of that 50% of the central nervous system anatomy. You're right. We looked at the cerebellum, we looked at the cerebrum, we looked at the spinal cord, we looked at the cortex, we looked at spinal cord smears, we looked at the dorsal root ganglion. Absolutely, right. This was big on identifying neurons, specific neurons, and then their locations and their structural classifications and their functional classifications. Absolutely. So that was a big part of that. That was a big part of that handout, and absolutely that histology handout. And I guarantee that's going to be a big part of the lab exam. Yes, but those histology slides, and there are a lot of them, are the only things that you're going to be responsible for histologically on this. Absolutely. So, but it is a good, I mean, it's more than we did in the muscles and more than we did on the skin, on the bones. So, I mean, it's the second most amount of histology that we've had. Not as much as the first test, but definitely more than the second and the third. Great questions. What's the next one? Is that it? Are we out of questions? That is an excellent question. If you are going, so, okay, we do indeed have a month break. So the first thing I would encourage you to do is take at least a good 5 or 6 hours off. Just take 5 hours where you're just not doing anything for, and then dive right into the new material. So here's what I would say. One of the nice things about 430 is it's very linear in the way it sets up. We have to set the groundwork first. So everybody who teaches 430 talks terms, anatomical terms and introduction first, talks chemistry, talks cells, works their way up. If there's any change in 430, some people like to do the nervous system before the muscle system. Some people like to do the muscle system before the nervous system. So again, there is very little variety in how you teach 430. 431 is very different. Every teacher has their own way that they like to, because each section is kind of independent of its own. They don't layer on top of each other the same way that 430 does. So if you are taking me for 431, we are going to start with the cardiovascular system, and we are going to start with the heart. That is going to be the very first thing we are going to do. If you take somebody else, they may start with other organ systems. I know several instructors start with reproductive. I know one instructor who starts with the endocrine first. Again, there's going to be differences on that. So what I would encourage you to do is to reach out to whoever your 431 instructor is and ask them for that information, where to start, and that would help you to get started on that. But if you're taking 431 with me, and I'll be honest, I haven't looked at the schedule, so I don't know who signed up for me or not. But if you're taking it from me, we're starting with the cardiovascular system and the heart. The other thing that I will do, and again, I can't speak to the other instructors, but because I think it was very important and very successful for summer, is like we did for summer, I will be opening up my Canvas site a week before classes begin. So again, you can come in and jump through all of the hoops. And again, even if you have me all the sections close quickly. But what I will tell you, Nicole, is that as grades come out for 430, there's going to be a lot of people who signed up for 431 who, as you guys have seen, I mean, look, we may have 20 students enrolled in this class, but we had 16 people show up for class today. So, well, actually, we started with 32. We started with 32, and now we are down to 16. So we literally lost half the class. And some of those people are still signed up for 431. So when grades come out, there will be a huge turnover. Usually the bottleneck is 430. So usually if you're trying to get into my 431 class, what I would encourage you to do is keep looking at that sign up, especially once grades come out for summer. Once grades come out for summer, there should be a huge turnover. I don't know how many of you were paying attention during the first week. That first week before summer, the first week before summer, class started. When I opened up the Canvas class, we lost 20 students in that week before class started. There were 20 students who were enrolled in the class and then ended up seeing how much work it was going to be and dropped. So if you get onto the wait list, if you keep checking every day, even up to the day before, keep checking on it to get into there. Especially if you're taking my class, even if it's the day before, the advantage of is you know what's expected of you, and so you can be able to get in that way. So definitely keep checking, and if you don't get on the wait list, then contact me, and I would be willing to add you to the wait list once the class opens, or not wait list, but I could add you to the class so that you could get in to see if there is availability depending on what's there. Since you guys had me for 430, I know you understand what my rules are, so I can be more lenient on adding you guys the day of the class or even the second day of class, because I know you know what to expect. But if I'm going to add you the second day like Sandy was there for the first day, got the lecture, got all the information, right, was there and active in that process and was able to be added that way. So I'm not just randomly picking people off the streets and adding them 2 days into summer school. I added people who were here, who were active, who were doing the things necessary. So as long as you're here active and doing those things necessary, which if you get in the wait list, you'll definitely be able to do. But again, the wait list will definitely open up. There's going to be a huge turnover in this. So if you're diligent and checking, you should be able to get in. Again, they're closed now because the wait list is full. But when summer grades come out, a bunch of people are going to drop or dump. Once my first email goes out reminding people that this is going to be a synchronous class, because even though it says it in the schedule, there are going to be people who are going to think, oh, it's online, it's not synchronous. I can go or listen whenever I want. As soon as they get that first email from me saying that, no, you have to be here at 8 o'clock in the morning, they're going to be like, holy crap, I'm not doing that. And they're going to be gone, or whatever it is. I don't remember. Summer, no, actually. Summer, it's the afternoon, isn't it? Isn't my 431 in the afternoon? I don't even know right now. I have one morning class and one afternoon class. But the point is, as soon as people learn that they actually have to be here on time, that is going to affect their desire to be in there. Once they get into the Canvas site and they see how much work there is in the first couple weeks and how many hoops they're going to go through, or once they learn that they have to take the exam on Proctorio, there's going to be a lot of people who aren't going to want to hassle with that, and there's going to be a ton of drops. In the first, like I said, in the first week, we had 20 people drop before class even began. From when I opened up the Canvas site to the first day of class, we had 20 people who were enrolled in the class who dropped. And on the first day, we had people who didn't show up. We had people from the wait list who didn't show up on the first day. And again, I didn't know if it was just me. I didn't know if this was online. I've talked to several instructors who teach online classes, and they say all online classes are like this. Because it's so easy to sign up for an online class, but then once you realize that it's actual work, people aren't willing to commit to that. So what I would say is that I would be, again, I'm not saying you have to take me, but you have learned what's expected of me. I would be shocked if, even if none of the 16 of you are on my wait list, I would be shocked if when it came around to fall, if you wanted my class, that we wouldn't be able to get you in. So keep watching the wait list, get on that wait list. There may not be any movement for the next week, but after the next week, once grades come out, once instructors like me can start sending out emails saying, hey, you know, people who are signed up for my class, this is going to be what's expected of you, you're going to see a lot of turnover. So spots will definitely open up in the month between now and the beginning of class. So keep being diligent up to the week before, up to the day before. Keep checking up to the day before classes begin. And if you get on that wait list, I will let you in so you can come that first day, and hopefully we'll be able to add you. So again, it shouldn't be an issue. All right. Excellent. Any other questions? All right. Well, in that case, like I said, I wish you guys tremendous amount of luck. Don't take this personally, but I hope to not hear from you on Thursday and Friday. Again, so hopefully there won't be any problems with the exams, but obviously I will be around. Oh, wait, for the final, what figures should we expect? I don't know. Yeah, I mean, so figures in like, so they're on, so it's not going to be lab exam type figures like that. But on that last daily quiz, there were a couple of figures on those where you had to identify certain features that were looked, you know, pointed at. So, you know, maybe bones, maybe muscles, maybe cell parts, something like that. There may be a few pictures like that. Yeah, like what is this feature or what is the structure? Those kind of questions, exactly. Identify this thing, a couple of those. But I wouldn't imagine there would be more than, you know, 3 or 4 of those types of questions out of the 100. So I don't think it should be too much of that. Right, but again, remember, even with be careful with the descriptive question, right? Because again, remember, they're all multiple choice questions. So I may have an arrow pointing at something on a picture and say, what is this structure? Right? Or what is the function of this structure or something along those lines? Or yeah, like what is this organelle or something along those lines? So yeah, so maybe some of those type. But again, remember, you're going to have the correct, there's still going to be multiple choice questions. The only other type of multiple choice question that you haven't run into before that you might run into on this exam will be processes. Because obviously before, if I wanted a process, you would write that process out. So what instead is I may give you the 4 steps of how a bone fracture heals, right? And so there'll be the 4 processes, and then all the multiple choice questions will be you put them in order, right? So it's 3 and then 2 and then 4 and then 1 or something like that. Or is it 1 and 4 and 2 and 3, right? And so you'll have to pick what the correct order is that way. So I think that would be the only other type of multiple choice question you haven't seen before that you might run into here. Otherwise, it's they're like the ones we've used on previous. It's not going to be the same ones. There may be 1 or 2 that are the same, but for the most part, they are novel new questions that haven't been used on the previous exams. And again, it's all multiple choice. All the correct answers are on that final. You just have to recognize the correct answer. All right, that's going to be a good job. You have no spelling, no pulling information out of the ether. You just have to recognize the correct answer. All right, I will be around tomorrow. So if you have any questions, any other questions before the exam, please feel free to let me know that. If you have any questions, yeah, absolutely. I agree with Michelle. Good luck to all of you. And if you have any problems, as always, please text me and let me know. Not text me, email me and let me know, and we will get that taken care of. Thank you guys very much. I appreciate it. All right, good luck. Have fun.